So good afternoon, everyone. Welcome to this edition of the NITheCS Colloquium. My name is Francesco Petruccione. I'm still the interim director of NITheCS. And today we have the pleasure of welcoming you, the people physically present here in this venue and the participants online, to a talk by Professor Inger Fabris-Rotelli. Inger is a professor in the Department of Statistics at the University of Pretoria, and amongst her many accolades, I just want to mention two. She's at the moment the president of SASA, the South African Statistical Association, and the CEO of the Institute of Certified and Chartered Statisticians in South Africa. Yeah. Inga, we are very glad that you agreed to join us this afternoon, and you're most welcome to start sharing your presentation. And while you do that, I will remind the participants online to use the Q&A facility to ask questions, and maybe at the end of the talk, we can give them the right to ask a question. Yes, so thank you very much, Inga.
Thank you, Prof. Francesco. I really appreciate it. And thank you, everyone, for having me. I hope my sound is okay. You'll let me know if you can't hear me or if there's any problems. Right, so this afternoon I have the pleasure of unfortunately not being in Stellenbosch and being in Pretoria, but at least we can do this online. So I hope everyone in the room is looking forward to it, and I am jealous that I'm not there in Stellenbosch. So this is I'm going to give you a talk about some of the work that I've been doing in spatial epidemiology, and you'll see just by my first slide that it's by no means only my work. We have a large team, and there's quite a few names there, and there's actually quite a few other names who I'll mention as we go along as well, who are also part of the team. So a little bit of background. So I actually studied applied mathematics at the University of Pretoria, but I specialized in image processing and applied maths, but I've always worked in the stats department. I majored in statistics as well as mathematics in my undergrad and honors. And then from my PhD in applied maths, I moved sort of into more statistical image processing, and remote sensing and spatial analysis is very much sort of the combination of those two things. So it felt like quite a natural home for me to fit in. So I'm very passionate about spatial statistics and spatial analysis and image processing. So during 2020, this team that you see here on the board on the screen got together to do some COVID modeling. And we all sort of knew each other before, but we actually hadn't done any work together because we're all from all over different institutions. So in a way, due to even though the pandemic was a very horrid time, it brought together researchers that otherwise would not have done any research together. So I'm going to give you a sort of a snippet of all the work we've done and we're still busy with. And I hope what you take from it is the possibilities of collaborative research across institutions and how productive it can be. So I'll get back to all the names a little bit later in my presentation so that you can meet them all. So just one introduction slide is on spatial statistics if you haven't seen spatial statistics before. So obviously we have a big field of statistics, and spatial statistics is one of our specializations within there. And the big thing about spatial statistics is all our data is now correlated in space. So traditional statistics relies a lot, not always, but a lot on our data being independent and uses this in our modeling. So spatial data inherently is dependent. So you have to take into account this dependency in some way. So there's a famous first law of geography by Tobler, and the reference paper is down there at the bottom, you can see it, called which states all things are related, but near things are more related than distant things. Very straightforward, very obvious, but this is the basis of all spatial analysis. We have to, before we do any modeling on data that has any spatial element to it, we have to understand how it is dependent. What is the autocorrelation in the data, and how are we going to model it? And then you feed that into other more traditional models, but taking into account the spatial autocorrelation. So there are four main types of spatial data that you might come across, and for sure you've seen them already. As I describe them, you'll realize what they are. The first is geostatistical data. So geostatistical data is our traditional spatial statistical data. It originated from mining in South Africa. And in fact, spatial statistics originated in South Africa. So it's one of the fields that we can take credit for. Because we were doing mining many, many years ago, Donnie Krieger, who invented a technique called kriging, which is famous in spatial statistics, was trying to predict where you should mine. So you would take certain mine digs and get a sample, and then you do sort of a fancy interpolation called kriging to predict where actually the best spot is. So geostatistical is that type of data. It's data from a continuous plane, but you've got samples at some discrete location, and you've got a value attached to it. And the question you're trying to answer is, can I make a continuous map from these point locations of data that I've got, taking into account the spatial autocorrelation? The second type of data is called point pattern data, which looks very similar, but what we're trying to answer is very different. So point pattern data is also points in a continuous plane, but we're not particularly interested in what the value is at the point, but actually why the point is where it is. So are there more points that are clustered together, further apart? Do they always sort of repel each other? Or is there actually no spatial autocorrelation present in the way these points are sitting in space at all? So that's a different question we're answering. The third type that you will have seen very, very often is called lattice data. So you get irregular as well as regular lattice data. Regular lattice is just image data, so it's just a matrix of pixels, and that's irregular because they're all the same size and they're left and right, they're lined up nicely. Irregular lattice data is things like our province boundaries or our ward boundaries or our country boundaries. So actually lattice data is a whole collection of polygons. So they're stored as polygons if you work with them in GIS software or in R. And there the modeling is different because there you have an aggregated value that is allocated to a spatial area. And now you want to see if there's you want to answer similar questions to geostatistical, but with this aggregated data. Sometimes it's called aerial data, lattice data. And then the fourth one that we work a lot with is linear networks. So linear networks are essentially just graphs, but we model them in a spatial way. In other words, we take into account the autocorrelation present in the nodes of this graph and where the edges are. And the easiest one example to think about is roads. So roads form a linear network, and you can model them as a graph, but then we want to model them spatially. You could also consider rivers. Another example of not a network, but a line pattern. So instead of point pattern, you could have a line pattern. And that could be, for example, fault lines, earthquake fault lines. Plate fault lines, not earthquake fault lines. So those are the four main types of data. But what's really key to all of these is that there's some spatial dependency present in the data. And if you don't take that into account when you're modeling, you're going to get the modeling wrong. So the first bit of work that this group of mine did was a spatial SEIR model for COVID-19 in South Africa. So we published this last year. This is a testament to how long it takes to publish things in statistics. We did the work in 2020, and we took a while to be able to get it published. It was actually accepted already in 2021, but took a long time to appear. So this is our team. So Pravesh Deba from the CSR actually started the research team. So he was the leader and the instigator and bringing everyone together. Jenny is with him at CSR, and Nontombeko is also with him at CSR. And then Sally was at that point, not Sally, Elise was also, as you see in this paper, also at CSR at that point. Zaid actually was at the Foundation for Human Rights, but this model, the spatial SEIR model, was actually his initiation. So he was put on a task team to assist the government in modeling COVID in South Africa, and he came up with the spatial SEIR model, and then he sort of joined the whole team together to sort of do some experiments and make sure it was a sort of publishable type of thing. So it's all can actually be attributed to him. And then we've got a few people who are young statisticians in the country and growing. So Raisa got her PhD during this period. Charles is busy with his PhD. Renata is one of my PhD students. She will finish this year. Nada also got her PhD, I think at the beginning of this year, end of last year. she's at the MRC. Arnold was a master student of mine, and Sibu is at IBM, but she was also a colleague at CSR. But what brings all these people together is we're all sort of statisticians that sort of knew each other, all been working on some spatial stuff, but not actually talking to each other and doing research together. So this was a really nice opportunity to really concrete our collaboration relationships. So Sally is not a statistician. She's from Wits University, and she was brought in from the epidemiologist expertise on this paper. She's also really lovely to work with. So this was a really, really lovely team, and we still meet every week. We're still very collaborative. So let me tell you a little bit about this model. So what was really interesting in 2020 was that we realized that modeling something that was happening is actually incredibly difficult. I'm going to go to the last line here on my side. Post-modeling something, once you've got a data set and you understood what the outcome was, is very different to modeling as you go. We don't know next week if there's going to be a surge, if we're in a wave, out a wave. What new thing are we going to learn about the disease this week, next week? How many people are asymptomatic? Is that really a thing? You remember all the questions and uncertainties that were flying around in 2020. So to model something like COVID in South Africa became actually a very interesting problem because we're very different from first world, from Europe, from the US. We are large, we're diverse, we have marked income inequalities and differences in housing, basic municipal services, transportation, medical care, and of course we're all aware that we have a very increased amount of morbidity risks due to TB and HIV. And all of this obviously really got us worried when COVID hit the world, and we knew at some point it was going to hit us. So all of this means that we have very spatially, and I highlight there spatially, diverse levels of vulnerability to the pandemic. And if you don't take this into account when you're modeling, you're not going to model accurately. So number one was we have heterogeneity in the COVID-19 prevalence. There's many factors, and it's diverse, and very important, it's spatially diverse. So it's different across the different countries, across within South Africa, different provinces, different wards, everything like all those spatial areas. An SEIR model with a spatial spread component is what we derived in this paper. So SEIR models are traditionally very homogeneous, and they don't take into account diversity and heterogeneity. So we included 4 infectious components for COVID-19 because of its complexity. We also included a spatial vulnerability index, and this was put together by Elise Leroux and her team. So there's a separate paper, which we'll reference later, so this was derived also in response as a request from the government, and it's based on socioeconomic as well as health susceptibility. So it's not just where you are and what your environment is like, it's also about the health in your area. And as I said, it was quite a challenging modeling exercise. Right, so let me step one step backwards. So the SEIR model is called, well, if I spell it out, susceptible, exposed, infectious, recovered compartmental model. And it's a very well-known epidemiological model, and you're basically following numbers of people in each of the compartments. But what's really underlying here in the base model is that we assume homogeneous mixing. So everyone has equally likely exposure to everyone else. And obviously with COVID, and especially in South Africa, this was not true. There was different levels of COVID, and from extreme to asymptomatic. And in addition, there's different exposure in terms of vulnerability. And we had to take this into account if we wanted to do the modeling. Also, epidemics exhibit spatial structure. They are spreading in space amongst the population. So it makes a lot of sense to be building a spatial model, not just a traditional SEIR model. And what was really interesting, I don't know if you remember, I'm sure you will when I say it, during the pandemic in 2020, when we were all locked down, you would see things on social media about cities in Europe, which are now ghost towns, and all the wild animals are now walking through the city and things like that. I mean, we didn't have that in South Africa. People could not lock down. It wasn't possible. So to model, so you could take an SEIR model and model it in certain areas. So this ward completely independently and just track what's happening in each ward. But because of all the movement happening in South Africa and the inability of most of our population to self-isolate and to not expose each other, you can't. So you had to take into account how people were moving and make this a spatial model. So this is what we did. So this is what the model looks like. So it's an SEIR model, right? In this model, we assume that the dead are in there. Okay, so some people will call it removed instead of recovered. But just to acknowledge that in here are dead as well, because it was very difficult to know the numbers that were coming through this due to cases made available, etc. But what was interesting, what was different in our models, we have these 4 components for infected, from asymptomatic, mild, severe, and critical. We assume that for certain percentages of the severe and critical would be hospitalized, certain percentage would die. Obviously, we're not specifying numbers here. And then these deaths would be included in this compartment. So what makes this spatial is all these little subscripts I that you see. So I is the wards in South Africa. So we ran this model at ward level. So there was a model like this that ran at ward level in South Africa for each day. So it was a time frame of a day, so discrete. And then, but at the end of each time period, we would, according to human mobility data that we had, we would distribute cases from exposed into neighboring wards, or even not even neighboring wards, further wards, based on the mobility that we knew about the populations within wards across the country. And then you would next day run everything again and redistribute cases. So we actually allowed for movement of all the cases across the whole of South Africa. So this model was run on a high performance computer, because you can see it's quite computational because of the high spatial resolution we're running at, and we were able to get some results. So I'll show you some results just now, but let me just give you a little bit more detail about the vulnerability index. So this vulnerability index from Leroux et al. Was published in 2020. It's a chapter in a special book published that year. It was published in 2020. And they were asked to put this specifically in place. It's done at ward level so that you've got a vulnerability index for each ward in South Africa. So it's based on transmission potential as well as health susceptibility. So transmission potential is based on the potential to apply social distancing. So this is about what houses are people living in, hand washing, good basic hygiene, etc. Health susceptibility accounted for older populations, higher disease burden, inadequate access to medical care, etc. So it had a lot of factors in it, and a really well-developed index. And this is a freely available index. If you Google it, South African Vulnerability Index, you'll find the dashboard for it. So this was taken into account in our model, in the spreading R0 value in our model, how it spreads out to, we scaled the R0 value based on the vulnerability index, so it was incorporated. So just to highlight again how diverse the country is, this is the vulnerability index per ward in South Africa, and you can see there's for sure no specific clusters. You can see sort of the high areas, so obviously a percentage ranging from 0 to 100, but enormously diverse across the country. So very important that this is taken into account when you do a spatial model. In our model, we also took into account age, and this was important because we have a very young population in South Africa. So I know you can't see the, apologies for that. This is the 0 to 19. This is the 20 to 59 age category, and this is the 60 plus, right? And you can see how much of our population is, and these are percentages in each ward, are young, medium, middle-aged, and oldish. So very, very much lower in the older age groups. So this was also incorporated in our model when we were distributing cases and in the vulnerability. Okay, so when we realized that modeling on the fly versus modeling once I had all the information was very difficult, we said, okay, the best we can do, we're never going to be able to match this dotted blue line here with the actual cases in 2020 from June to December, because there's so many unknowns in there. Number one, we know that not all the cases were captured. Not everyone got tested. Often our whole household would get sick, but only one individual would get tested, etc. So it's very underreported. So we thought, okay, the least you can do is just argue that the model is doing more or is doing better than if you just did a standard SEIR. So in this figure, the red line is the cases from our model, and the blue line is a national model. In other words, I didn't do any mobility between wards. We just ran the model on South Africa for the cases and let it distribute out. So we obviously started here at the same point and we distributed out. And you can see how massively over predicting it is. Obviously, one could argue maybe we missed that many cases, but I don't think so. The red line is the cases from our spatial model. So this model is, so we ran our model at the ward level and then we aggregated it up. But we did the running of the model spatially. So we allowed for spatial dimension in the implementation. And you can see that this is, first of all, not overestimating as much like that. And it's also capturing the curve better. Of course, it's not on the blue line, but that is because of our, these are confirmed cases and not perhaps the true cases. But the point is that it's definitely doing a better job than the green line, that if you don't model spatially. The other thing to consider was the choice of R0. This is a parameter that we need to estimate in SIER models or epidemiological models. And this was actually a very interesting exercise. So we actually have an unpublished paper, which my PhD student Renata did in 2020, on the uncertainty of this R0 value across the world in the first 6 months of the pandemic, so from December to June. So she did a giant literature review on all the different R0 values that were popping up all over the world. And she had a big, once she'd done the literature review, she had a nice discussion about how much it's varying spatially. And a lot of it was to do with where the severity of the cases were. So remember, it was China, it was Italy, and there the R0 was sky high. And South Africa was much more conservative. It was very, very interesting. But unfortunately, as is the nature of publishing papers, we submitted that paper and they sat on the paper for 18 months until the end of 2021. And then a review came back and they said, oh, but why did you only look at the first 6 months? You know, the stuff's so old. And I wanted to say, well, if you don't review it in time, that's what happens. So needless to say, the paper is unpublished, but it's on archive if you want to read it. Right, so we then tested the choice of R0, knowing that this is a parameter that needs to be considered. So NICD did release possible values of R0 in different provinces, which was very well done. So we tested it in our model, the various values, and you can see that this 2.4 value at least captured the slope quite correctly, but probably the peak not well enough. But of course, once again, the cases are underestimated. So this is a difficult parameter to choose or to justify. So a lot of the modeling that we did in this paper that was published was based on literature. Parameters were chosen based on literature, including these parameters, so infection rates, incubation rate, all of those sort of things. So we didn't thumb suck any of them. Based on literature, we have a nice table in the paper which justify how to choose them. Okay, so once we had scaled R0, so we're going to choose an initial R0, all right, based on literature and on some simulations. So 2.4 seemed like a good choice. Now you want to make sure that you've done the right decision, because what we're doing here is scaling this R0. So in other words, more vulnerable populations have a higher R0 than others. So we want to scale it based on that vulnerability index. So to test that what we did was correct, once we'd run the model, so this proportion of infected individuals is from our model, we check the correlation of that between R0 and what our model output gave. And you can see that most of the correlations are very high, so larger than 0.6. Some of them didn't work out. There's a few little gray ones, but not many. That's just because there was no data in those areas. But a lot of them are very high. But you can see that the bigger areas in our Northern Cape have much higher values, for example, than down here on the coast, but still mostly high correlation. So this is sort of a validation. Okay, the model's at least doing the thing you want it to do. Right? It's going in the right direction. Right. What was really important, obviously, during the pandemic was hospitalizations. So we did some simulations, again, for each of the provinces. So we, again, ran this at ward level, but then we aggregated it up to the provincial level to report. And this was because we realized even the case data that we had. So we were made available because we were working with SIER and the NICD. Case data at ward level was made available to us, which was quite confidential at that point. But we realized that this was very uncertain data. So a lot of the cases that were reported that were allocated to a certain ward were not the correct address. It was often the address of the doctor's offices, and the individual's home address didn't go onto the test, or individuals would go to other places to get tested because of stigma involved, so their true addresses are never on it. A lot of people got hospitalized during 2020 and 2021 and were unconscious, so you don't actually know where they come from, so the hospital area is allocated there. So very uncertain. But if you aggregate it up to, once you've done your running of your model and you aggregate it up to provincial level, then you can, that maybe that uncertainty has been removed. So we're able to do hospitalization simulations, and this matched quite closely to the true data. So there's obviously a lot more in the paper, but I'm not going to show you all of that because I want to show you a snippet of everything we've done. What was really important, we had lots of discussions about these papers. In the first half of 2020, we were meeting twice a week online and discussing these things because we really were excited about being able to do something and hopefully contribute to our fight against COVID in 2020. Our meetings were 8, 9 o'clock at night because some people had babies and then the babies were sleeping. Just some of us were homeschooling. It was an interesting time. But we had lots of conversations there and really nice in-depth modeling discussions. So the model parameters I already mentioned before, that's something that was very difficult for a disease that's unknown. It's much easier when you have data, historical data, and you know some things about this disease. Every day there was a new story about how many people are asymptomatic, is it really a thing, what's the rate of infection, etc. So in the end, we have a very nice table from literature on choices that we made. And some of the model parameters, we even gave distribution so that we didn't have to pick a specific value. We sort of gave a range and we said, there's uncertainty involved, and that could be included into the model. So it has some stochasticity in the model. The other thing that's very difficult to model is how the society changes when there's intervention. So we had steps in the model which took into account interventions, government interventions. We could change a certain scaling rate, but you don't know how the population themselves are going to react to it. So that's very difficult to take into account. We were also, as you saw in our picture earlier, we needed information on how people are moving between wards. And again, we were given data from Vodacom at ward level, proportions of people each day that move between wards, cell phone data, very confidential. Obviously, it was not, didn't have any personal information in it, but not the data that a cell phone company would share under normal circumstances. And unfortunately, this was not available after June, so we only had a certain amount of it, but we could at least fit the model for the first half of 2020. I already spoke about case data location accuracy. That was very tough to deal with, but we made the decision that we model at ward level because you can capture a lot of heterogeneity. We have mobility and we had actual case data at these two levels. So if we could at least model it there to bring in the heterogeneity and the spatial information that's happening and then report it at a higher level, we're in a good spot. Of course, at that point in 2020, we knew very little about immunity, if someone could get sick in the next wave, all those sort of things. And of course, we didn't have any vaccinations yet, so that was not in our model. So the next paper we did was about the mobility, because we realized that this was quite complicated. And when our cell phone data source seemed to disappear, we didn't have any mobility data. Now we couldn't do any more modeling. So Arnold was a master student of mine at the time, and he did some very, very nice work on how you can model mobility. So there is data on mobility available freely to the public from Facebook and Google, but this data is only available at district level, not at ward level. So it's at a much lower spatial resolution. And if you want to model high spatial resolution and really capture the nuances in modeling spatially in South Africa, this is not good enough. So in this paper, which we published in 2021, before we even published the other one, even though the work was done afterwards, we looked at how you model mobility. So you need mobility, or you need some sort of spatial information between the wards. Okay, and this in this case for COVID was human mobility. Okay, in spatial analysis we capture this with a spatial weight matrix, and specifically in this application we call this a spatial mobility matrix. So a spatial weight matrix is just a matrix. Let's say there are 1, 2, 3, 4 up to little n spatial areas, so then it's an n by n matrix, and this weight wi is just the spatial weight between unit I and unit j. Okay, if it's mobility, it will be the proportion moving from this unit I to this unit j, for example. So traditionally, these weights have to sum to 1 to make it a proportion, but this doesn't have to be so depending on your modeling situation. But basically, this weight quantifies the amount of spatial influence that spatial unit I exerts on spatial unit j, and in our case, mobility. So for example, if you look at Gauteng and our 5 districts, you could have something like this. So what's nice is you can actually represent it as a graph, right? So edges between the centroids in the 5 districts here, and then you've got some weights here. Okay, and you'll note in this case, they're not standardized, and they're not summing to 1. But you can see there's a lot of movement from Joburg to Tshwane, but from Tshwane to Joburg, not so much. So this matrix is not symmetric. Some modeling in spatial statistics wants this matrix to be symmetric, but we're specifically looking at mobility, so it's a directed type of matrix, and therefore a directed graph. Okay, so as I said, there's some other data available. The one is Facebook for good data. This is freely available, but it's only available at this level. So these 5 districts. There are many, many more wards in Gauteng that you would like to be able to model at. So this is just a district municipality level. This is what the mobility, Facebook mobility data looks like. So this is 2020. You'll see in March. So the naught is like a baseline. So Facebook models it according to some baseline. You can see the index FI T is between -1 and 1. So I is the spatial unit and T is the day, right? And then there's some baseline week, which was Feb 2020, so before the pandemic began. And then they looked at mobility relative to that, right? So it's not the same type of data as cell phone data, because there we had actual people going from I to J. Here we only have a relative value just for unit I. But you can see for sure that the mobility, so the red is the average mobility, dropped immensely in March when we locked down, and then gradually obviously increased a bit. And the green here is our COVID cases. So as our COVID cases increased, so this was obviously locked down and we didn't see cases for a bit, but there was definitely an increase. The cell phone data we had was very interesting. So we had it from March until I think the end of May, just in June. And you saw this was before we went into lockdown. So this is all the still normal, and then a very sharp drop in mobility according to cell phone data, and then it stayed pretty low. So we had this data for the first half of 2020. We wanted to continue modeling. We no longer have it. How do we use this? Right. So Arnold did a nice study here in this paper on what are these different data sources telling us, and number one, how do we make them comparable? Right. So we didn't look at the Google data in this paper. We just look at Facebook and the mobile network data. If you want to compare them, you want to have it the same spatial resolution. So we don't. I've got a master's student working on it now, but we don't have a technique yet to bring the Facebook data, which is at district municipality level, down to ward level. But you can obviously aggregate up from ward level to district municipality and compare. So that's what Arnold did in this paper. So it's not a simple aggregate, because remember, you've got links between wards, and you have to try and somehow aggregate. You're not summing. There's a bit of an algorithm behind in that process. So he worked on that. Then we looked at what is the best way to now create this mobility matrix that we want, because we need it in our SEIR model, spatial SEIR model. So the one way, which is very common in spatial statistics, if you've got, for example, something like this, the weight, this red line weight here will be the inverse of, well, it'll take the distance from this point to this point as the crow flies, right? And plug it into this equation. So you get sort of an inverse distance. Okay, so the closer they are, the higher the weight, the further are, the less the weight. So that's a simple one, straight distance method. But now we've got other data. Now we've got mobile network data. So how do we take that into account? So for the cellular data, we took the weight as the data we got from the network. The proportion of people moving from I to J, unit time, T. Okay. The Facebook data is more difficult because here we only have a single value, Fi of T. Okay, so how do you bring in Fi and Fj to get a weight? So we use a very nice formula by Irregular et al., which was a paper published in 2020 in Spatial Stats. This is probably the biggest spatial stats journal in the world, an Elsevier journal. And this weight matrix takes into account the distance between the centroids of the spatial areas, but then also an absolute value between the two mobility values we've got between I and J. And it weights here, you see it's a weight between the distance and the value. So it's not just a distance weight, but it takes into account a covariate, which in this case is F. Okay, and in our paper, we set this equal to 0.6. And then the other way was to take the distance one, but then scale it by how much, because we're going from I out of I. So the distance takes into account how I go out of I, but the Fi will be the relative movement in I, okay, whether it's up or down. And this was another way to do it. So just a summary of what his results did. So he had a nice simulation study and checked and some graphics and results. The distance, the first one is very simple. The second one is obviously very nice, high spatial resolution, but we don't have any data anymore. It's actually not representative, which was quite interesting, because not everyone has a cell phone or has their GPS on. So we had a lot of discussion about whether cell phone data is truly representative in a country like South Africa. On the other hand, Facebook apparently has massive penetration into an African market, like South Africa. So there's actually more representation from Facebook data than there is from, and this was Vodacom data, than from a network like Vodacom, who only holds, if I remember correctly, about 45% of the market. So this, unfortunately, is a low spatial resolution, but it actually captured quite nicely. So what was different between the Vodacom data allowed us to know the strong associations over the short distances, whereas the Facebook data told us more about the movement between Cape Town and Johannesburg and big movements like that. So if you want, there's some more paper there. What we're doing, what we're working on now is I have a master's student, because we realized you want this data at high resolution. So we're working on a novel kriging technique to actually take Facebook or even Google mobility data and small area estimate it down to ward level. And the value of this is that Facebook and Google data are publicly available, and it would mean that any country could technically, provided they have the right covariates, then have mobility data at a low resolution without worrying about the sensitivity of cellular data. So we're working on that still. Don't know. Next year, there'll be some information. All right, the next thing to take into account was obviously vaccinations. So Claudia Dresselhaus, also a master's student of mine who finished last year, graduated in April this year, worked on this. So she took our spatial model from before and added in a vaccination component, and you can see the same team there. Warren from Nelson Mandela also joined us in this project. So this looks very similar to before, except that we've added in a vaccination component of a compartment here. And we then split our infected compartments slightly differently, because now we understood the disease better. So we now realize that if you're going to be asymptomatic, you're going to stay asymptomatic. But eventually, if you're going to get sick, you will be mild, and you will then progress to severe and critical. People don't go directly to critical, usually. And if you do, it'll take into account these rates. Maybe this will be shorter, type of thing. Okay. So we adjusted the model, and then we reran some simulations. So in this paper, which is currently under review, we ran it only on Gauteng, because Gauteng gave out nice vaccination data per district municipality. So again, we're only at the 5 district municipality levels, because we don't have mobility data at a higher resolution. And she conducted a sensitivity study to see if the model that we've set up, so similar to what I showed you earlier in the slides, in fact, is doing what you would like it to do. So you're not, it's not contradicting you. So the parameters that came out really strongly in the sensitivity analysis were initial immunity, basic reproductive number, lockdown scaling, and percentage asymptomatic, and a little bit here, mild hospitalizations. And that seems very logical, much less than if you look at what's going on here later on. So model is not a bad model. Of course, then you want to check that it actually fits what's happening. So the solid lines here are the actual cases. So here we've got our 4 waves, and a small little 5th wave over there, which we know wasn't really significant. And then our model in the dotted line with confidence bounds here as well. And you can see in the beginning, the confidence was wild, and then it sort of calmed down. But it's definitely capturing where our waves are and telling the same story. Again, the actual cases were highly underestimated. So we looked also at the hospitalization cases, and here you can see our solid and our dotted lines are really telling a good story, because the hospitalization data is known. You knew the person was in hospital. There's still a little bit of uncertainty, of course, because sometimes they won't be reported as having COVID if they never did a COVID test. But generally, that's why you get the uncertainty here. But later on with the smaller waves, obviously there's more certainty involved in those. So what we're working on next is an age stratified model. So I know I said that we included age earlier, but we didn't include how people interact between age groups, and we only had 3 age groups there. So we're working on a much more in-depth model on different age groups and how they interact with each other. So again, adapting our spatial SEIR model. We are also working on a small area estimation to get high resolution mobility data, and we're hoping that this can be a model that can be used all over the world if you've got Facebook mobility data. I'm also involved in a project in strengthening women's research network and capacity to address women's health in sub-Saharan Africa. A group of, so Nada, who's my collaborator in this SEPIMOD group, and some other ladies from MRC and Malawi and someone in the US have got a nice, quite a good grant, and we're starting this at the beginning of next year. And Nada and my roles in this is very much about the spatial part of it, which is really, really nice. We're also working on disjoint spatial lattice data. So in the examples that I showed you, so maybe you want to consider Gauteng, you've got it's all continuous. All the areas are touching. There's no holes or gaps in your areas. But you could have data which is collected only in this area and they're disjoint. And in spatial modeling, this is not normally the case. Normally you have your continuous lattice data and everything's perfect. So I've got a PhD student who's also working on that and how you model COVID and accessibility in that sense. And then what we're really working on now as a team is wastewater modeling, because the pandemic is over. So what's next? How do we be prepared for the next pandemic? And wastewater modeling is a really nice alternative to that. So we're working with some additional researchers from CSIR. We've got wastewater samples from Irregular in South Africa. And this is part of a bigger project called SUCCESS, the SA Collaborative COVID-19 Environmental Surveillance System. And it has 87 wastewater treatment plants across South Africa. Our project with CSIR involves 5 to 8 of these sites, because some of them weren't sampled in the beginning, in Ekurhuleni from August 2020 to Feb 2023 at this stage. So wastewater sampling is a nice alternative to getting actual case data, because you test the entire community instead of individual-based testing and where you might miss tests. Everyone is using the water, so you know what's coming out of the water. So you just need to know, you need to have some idea of what area feeds into the wastewater plant, and then you can model like that. So they take samples and they extract RNAs, which is gene copies per milliliter. I know nothing about any of these sort of things, so I'm just putting it there. If you understand it, that's good. But it is quite complicated. So you get a lot of variability as well in this, as you did with the individual based, due to temperature, the time to the testing, the degradation of the sample, how often you sample. And what was very interesting, the data collectors was saying that weather is a big issue, you might not be able to get there, and social unrest, there might be protests and things like that. And then obviously you've got to take into account the wastewater treatment works size and the feeder areas. So there's this SUCCESS project has got a very nice dashboard that's really available to everyone. You're welcome to look at it. So this is Ekurhuleni. This is one of the district municipalities in Gauteng, so one of those 5 areas. And the colors that you see there, the gray is the areas which haven't got samples for, and the colors are the wastewater feeder areas. So you'll see the little pink dots, those are wastewater treatment plants. So this one, for example, flood plots, all this light blue feeds in there. Okay, but now if you look at it, you can see immediately the complication and the complexity in the modeling here. The light blue is totally disconnected. So these gray areas in between are fed by another wastewater plant, where we haven't done sampling. So this is the situation that we have. This is the spatial domain we're working on. It's not the nice Gauteng with everything continuous. It's Ekurhuleni, but we have very disjoint data. But what would we like to do? We'd like to be able to say, this is how much COVID's here in an area we haven't sampled. So this is what we're working on at the moment. So because you've got 2 spatial lattices here, you've got the colors, and then you've got the gray when you want to model. So you've got 2 spatial lattices that you need to consider. Normally it's 1 and you model on the same thing, right? So to do that, we have to have some measure of the relationship between these 2 spatial lattices. So let's assume they're P1 and P2 for polygon. Okay, so it's polygons 1, 1 up to polygons 1n and 2, 1 up to 2m. This is a very nice paper, very recent paper in Spatial Stats that suggested 3 possible distances. Obviously, you know, as you go through a paper, the last one is the most useful. The first one is just simply the distance between the polygons. So the shortest distance until they touch. Not very useful because it doesn't take into account at all the shape of the polygon. The Hausdorff distance is another one, a bit better, but still not allowing for the shape and how you take it into account. So in this paper, Gotti et al. Suggest an approach through Minkowski addition. So you pick a polygon in the one lattice and you enlarge it with a ball, okay, with your Minkowski addition until it covers the other one. So the other way around, yeah. And then the speed at which the one covers the other is your distance measure. And this was really nice because this ball is not forcing it to be a circle. It's keeping the shape of the polygon until it captures the other polygon. So what we're working on next is and this is a current project this year, so I haven't got any beautiful results for you yet. If you ask me in December, I'll have something for you. We want to use this distance measure, so the speed at which they cover each other. So then you can have a measure between every pair of polygons here, right? And put this into a weight matrix. This will obviously not be same number of rows, same number of columns. And then you use these weights in a spatial model. Okay, instead of the traditional weights matrix, you use these to get the relationship between the 2 lattices. So there's a number of different models spatial models that have been done. Bayesian hierarchical models is a very commonly used one, but this idea has not really been done at all in spatial analysis yet. And the other idea that we're looking at is taking our spatial SEIR model and putting this new weights matrix in instead of that mobility matrix and modeling like that. And that is what I have for you today. Thank you.
Yeah, thank you very much, Inga. The questions for Inga. Please.
Thank you very much. That was very interesting. I'm quite curious. You saw that correlation between you saw there was that spatial structure in the correlation between the Arnold values produced by the model and the what was it? I made a note and the vulnerability index that you add.
This one.
So, where you could see KwaZulu-Natal and like Cape Town's looking very different from the Northern Cape. And so do you have any explanations for why that might be? Do you have any hypotheses?
So just remember, maybe I didn't explain it properly. This is not the true cases. Here we're trying to check if our model is going in the right direction. So where there's a high R0 value, you want to see high case numbers. And where there's a lower R0 value, you'd like to see lower case numbers. So all this graph is a the the correlation between the R naught that we put into the model and the output of the model, making sure it's telling the same story. And most of the all of the correlations are higher than 0.6. The reason that I think that you see slightly lower, but not no correlation here, is because the spatial areas are so small. So because we were feeding the original case data into our model as starting values, and in the smaller areas, there's going to be much more variability than here. Because you see, these are all sort of, these areas are bigger, so there's going to be less issues of reporting the incorrect address when you do a COVID test. So whereas here, you're going to get more issues. So it's probably also got to do with the case reporting in maybe these poorer hospitals and poorer areas that have resulted in this sort of thing. But all the correlations are high, meaning if we chose the right R value, the cases that we simulated with our model told the same story. So it's a model validation approach, I suppose.
That makes perfect sense. Thank you.
Thank you.
More questions here? Otherwise, we can ask.
Hello?
Yeah, please.
Yes, sir.
Go ahead.
Okay.
Yeah, go ahead.
Since the issue of COVID is seasonal, I mean, it takes place during a certain period of time. Then there is issue of losing some instances that you need to make model for, like the streaming data, just because the case of a COVID situation is for that period of time. It may not come as soon as possible, and there are some decisions that needs to be taken at that right period. So are you thinking of going into issues like using streaming data to model some of the cases in case there is an issue like that comes up, so that people can easily take proactive action as soon as it comes in, rather than using the whole data set. Like now, what we are doing now is old data set, not when it comes in.
Absolutely. And I think this is some conversations we had definitely in our group. So that was what was so difficult. You know, the government in 2020 they had quite a few modelers, and we weren't even one of the big teams that were doing work. They wanted them to be able to say, what's going to happen next? Where should we go? And I mean, and here was a disease with variables that we really didn't understand. But that's the hope of what we're going to achieve with the wastewater. So that's why they put the surveillance system in place. So, and it's not just for COVID, it's for, remember we had a cholera outbreak a few months ago in Hammanskraal. If you have a proper wastewater sampling process in place with methodology already in place that we would like to develop to say, this is when you get this value coming out of here, this is with community that's at risk, go in there. So, and that's exactly what this wastewater project is for, because you can't, with case data, it's so delayed. You know, you'll by the time we got updated information, so we got, until January last year, we actually got hospitalization data from the Gauteng government, but we would get it every Monday. So now you've got a week's worth of data, and now you must process it within an hour and release it on a dashboard. I mean, it's full-time jobs. Whereas a wastewater process, if you've got the methodology in place with a dashboard, you can pick these things up much faster, and other diseases. The other thing that they're really interested here in wastewater is antibiotic resistance. They can pick it up in the wastewater. So very, very powerful way forward, I think, in terms of disease monitoring and tracking, wastewater collection. I hope that answers the question. It's very complicated.
Thank you.
Very complicated, as you said. So must think on that area, that streaming data. Maybe you look at that feed. Well, I can link you to people that are doing it, so that you can do it.
Okay, please. Thank you.
All right.
Okay, please. Thank you. Wonderful suggestion.
Thank you. There's one question at the back. Yeah. Question is coming.
Hello.
Hi.
With that wastewater modeling thing, the whole gray area, you guys said you didn't sample that area. Is it possible to get the data for that area? Because it seems like a large area as compared to the whole small area. Are you guys planning to sample that area, or is there a way to get data for that area?
So your question is that you're asking a statistician, would she like more data? And the answer is yes, always. So absolutely, the ideal situation was that every wastewater plant in South Africa is sampled once a week at minimum. I mean, obviously, maybe once a day, but once a week. But here we've got limited funding. So there's limited funding to be able to go. A human has to go out there and do the sampling. So it's very human intensive. So what they're doing, so they've chosen only 87 wastewater treatment plants across South Africa to do a proof of concept and prove that this is a good idea. All right. So it's very, as with all data collection, I mean, the only census we do is our national census. No one ever gets all the data for everything. So the idea of statistical modeling is always, based on the sample you have, can you estimate elsewhere? So instead of spending money to get more samples, which is amazing, but probably not sustainable, can we take the samples that we do have, these 8 sites, and then make a statistical model that will predict on the gray areas using other covert information? I mean, that's first prize. That's cheaper. Statisticians get something to do. Everyone's happy. More data is always great, but we're not going to get it, unfortunately.
Thank you.
Thank you. Mesias, please.
Hello. Thanks, Inge, for such an interesting talk. Have you looked at the stochastic modeling of, or you just consider a deterministic model?
For the wastewater?
No. In all your models.
The original model. So it is a stochastic model in some sense, in that some of the parameters are modeled with distributions to take into account the uncertainty. And we don't always do deterministic movement of cases. We have some probability in there, but we haven't done proper stochastic modeling of these. So we do have, so the model is represented by some PDEs in the paper, but actually that's just the representation of it. In reality, we're doing a computational simulation with the model. But I think there's, I gave this talk a while ago as well, and a mathematician also said to me, what about the stochastic proper modeling of this? And I think that's definitely something to look into for sure.
Yeah, thank you.
Inge, there doesn't seem to be any more urgent question. Maybe one last one here. Sorry.
Hi, Inge. So I suspect your model would be very sensitive to the spatial heterogeneity of your data sampling, because if different areas have different sampling rates, for example, that might skew how much your model think is happening in specific places. So how do you take that into account in your model?
Yeah, in the wastewater or in the original model?
Thinking about the original one, but I guess that also wastewater modeling.
So I think because we take into account so much heterogeneity already, so in this model, we because of the mobility, so like the heterogeneity in the case numbers is obviously that's the actual data. So you want the heterogeneity in the case numbers, but we do take into account the heterogeneity of the mobility of everybody, and through the vulnerability index, a number of social and health coverts. So that's taken into account there. For sure, there's probably more complex ways of bringing in this. You can make a model as complicated as you like, but you also want to be able to run it and get some reasonable results out. So, but absolutely, I think there's many more possibilities that can be done here.
So you're hoping that essentially by modeling your vulnerability, that will ultimately take into account the fact that your sampling of the data will differ between regions that is somehow correlated with vulnerability.
Yeah, yeah,
Okay, thank you.
exactly.
Okay, Inge, then now I think there are no more urgent questions. So thank you very much for your time.
Thank
for
you.
the talk. You heard there were lots of questions, and maybe in the near future you can give us an update talk to hear how these
Sure.
techniques evolve. Yeah, so